Good afternoon, everyone. So we're going to actually get started. I know folks are trickling in, but we have a lot of interesting talks for today. So I want to make sure the speakers have enough time to cover their content. So thank you for joining us for the last seminar of the semester. We'll be talking about Rhode Island AMIA Year in Review. It'll be presentations by students within our center. And then just want to let you know, we will be continuing the series in the spring and continue to do them bi-weekly Fridays at noon. So the next one will be on January 22nd, and we'll make sure that we share the schedule and make announcements with our usual mechanisms. And our speaker will be Dr. Kim from Harvard Medical School and the VA Boston Healthcare System, really talking about dissemination and implementation. So the plan for today, I'm just going to give a little bit of introduction. We thought this might be a great opportunity to just talk a little bit about AMIA, which is the professional home for biomedical informatics, and talk, as the end of the year, give a little bit of a year in review of what's been going on with AMIA this past year, in particular, what happened at the annual symposium in November, where we had some of our students present. And so you'll be hearing a little bit about their studies, as well as some highlights, some sessions they attended that they thought were really interesting and wanted to share with you. And so each of them will be talking, but to start with, thought I'd give an overview of what is AMIA. So AMIA is the American Medical Informatics Association, and so it's the professional home of biomedical informatics, as I mentioned. Actually was formed from the merger of 3 organizations about 30 years ago, as shown here. And there is kind of the honorific college called ACME, so the American College of Medical Informatics. And so as shown here, the AMIA mission is really to improve health through informatics, education, science, and practice. So quite broad, and kind of ties into what we talked about on the first session of the where we gave an introduction to biomedical informatics and how there's a lot of things that go into it, many subdisciplines, methods, applications, and really covering a full spectrum of research in informatics. And AMIA has a lot of different programs and services and really focused on advancing science, promoting education, and really advancing the profession. And we're really looking to kind of train the next generation pipeline of informaticians. And so in terms of education, AMIA runs a number of meetings throughout the year. And so as you may have noticed, as we kind of have these sessions, it's really a broad field. And so some of us, do some policy work. So there's a meeting focused on that. And there was one in 2019 focused on clinical decision support in the era of big data and machine learning. We have meetings dedicated to education and really education at all levels, high school, all the way to professional, and maybe someday it'll be even earlier than high school, but there are meetings for that. There's also meetings focused on in March, the Informatics Summit. So that's one of our newer conferences, really focused on talking about work in bioinformatics, clinical research informatics, implementation, and data science. And then in May, we have the Clinical Informatics Conference, which is much more on the applied side. And then finally, annually, we have our annual symposium, which is what we're going to talk about mostly today. So we just had that in November. And that's really kind of where we bring everyone together. And this is the largest meeting or conference for AMIA. And so you hear a little bit of everything, but it's a really nice way to kind of see what is going on in informatics, what's the latest, and then kind of see the work that's going on across the country as well as internationally. So just to kind of talk a little bit about the Virtual Annual Symposium. So this is kind of what it looked like when we were kind of going to the conference. And so this is our welcome, and we would go in. And just to give you a sense of what is covered in annual symposium is we have a range of sessions. So we have ones that have oral presentations. So there's usually a few of these per session, and these cover papers, student papers, as well as podium abstracts. We have poster sessions, we have panels, systems demonstrations, and then workshops, just again, covering a range of topics. There are a series of programs, and so I thought it would be nice to highlight some of these. Again, as we mentioned, education is really what we care a lot about, is really training the next generation, really having a pipeline of students really wanting to go into the field of informatics. And so several years ago, we started a high school scholars program. There's a first look program for undergraduate women. There's a student paper competition to really recognize top papers. Student design challenges. There's also career development for women AMIA, so those that kind of at the professional, but really throughout their career. And then also just wanted to mention this one. So we heard, I think, about Smart on Fire and Fire, I think a little bit at Dr. Fraser's presentation. So there's actually a competition now for different apps and kind of highlighting them and showing them. And then they kind of recognize the top apps that are out there. There is an opening and closing session. So I just wanted to highlight, our keynote speaker talked about the social dimensions of science, technology, and medicine. Our closing keynote was Dr. Denny, who's been in the field of informatics for some time. He's now the CEO of the All of Us program. So that's an effort to really recruit and create a database, a national database of 1 million or more individuals to really have a diverse database to study and improve health. So talked a little bit about what they've been doing, what both of these keynotes talked about what they've been doing and kind of, how informatics relates to that. Also wanted to just mention at the opening, this is a tradition, is to honor the Morris Collin Award of Excellence. So this is the highest honor in ACME. And so this went to Dr. Zach Kohani, who's actually a Brown alum. He's up at Harvard as a chair of their Department of Biomedical Informatics. And then he gave a nice talk about finding the doctor in biomedical informatics and really highlighting the importance of informatics' role in health and healthcare, and really the need to kind of be more engaged and involved, which was also a great thing to hear from him. Other feature, just wanted to mention quickly, is there was a nice fireside chat between Dr. Brennan, who's the director of the National Library of Medicine at the National Institutes of Health, that funds a lot of informatics research and develops resources like PubMed and GenBank that many of you may use on a regular basis. And so she gave a fireside chat with Dr. Fauci, kind of talking about the potential role of informatics. And then just to highlight here, the next day, you kind of highlight how he talked about the role of informatics, importance of data sharing, and then the development of registries to track patients receiving vaccine doses. So that was kind of a nice highlight from the conference. And then also every year we have a informatics year in review just to highlight things that are going on in informatics, what's the latest and greatest. And then so what this graphic shows is all the working groups in AMIA. So just also again to show you the diversity of what AMIA does. We have all these working groups and each of them selected some key papers to really highlight what's been going on, and then kind of showing how a lot of them have kind of contributed to responding to COVID and quickly kind of doing work in that space of using data or technology, indeed different methods in informatics to help understand what's been going on. And this, just to highlight again, was given by another Brown alum, Dr. Jim Cimino, who gives this every year. So those are just a couple of highlights, but just wanted, this session is really about what Rhode Island has role in AMIA. And so this is highlights 5 student papers. So there, I think the statistics are that there were 75 student papers submitted, 35 got accepted, and then 5 of them were actually from Brown, and really showcasing the variety of work that we're doing here in Rhode Island. And so that's what you'll be hearing about. So we'll be hearing a little bit about what Aaron's been doing, then followed by Jillian, Katie, and Ryan. Just to mention, Sotiris is another student. He graduated, he's now at Columbia, and so he did some work with natural language processing with our data from our health information exchange here in Rhode Island, Current Care. Also our faculty, so Dr. Fraser, who we heard from in November, gave a workshop on global health informatics, and our other faculty, Dr. Eichhoff, who we heard from students and faculty in his lab, some of the work that was initially accepted to the Informatics Summit that I mentioned in early March, but they had the opportunity to present their work at the symposium. So some of the work that they're doing with machine learning and diagnostic decision support. So with that, I will hand it over to Aaron, who will talk with us a little bit about his study and then some highlights, some of the sessions that he attended that we wanted to share with you.
Thank you for that introduction. Today I'm going to give an abbreviated presentation of the talk I gave at AMIA a few weeks ago on our paper, Extracting Angina Symptoms from Clinical Notes Using Pre-trained Transformer Architectures. Cardiovascular diseases are the leading cause of morbidity and mortality in developed countries. The primary mechanism for cardiovascular disease is the development of atherosclerosis. This results in a narrowing of the lumen, increased risk of plaque rupture, and downstream total arterial occlusion. When this occurs in the heart, it's known as a myocardial infarction. Complete elimination of blood flow for an extended period of time leads to cell death. Depending on the size of the affected tissue, this can result in irreversible heart failure, functional impairment, or death. It is common for older adults to have highly occluded arteries. These obstructions can be detected with a range of tests. Treatment options include both medical therapies and intervention. Clinically, the goal and challenge is to identify patients who have underlying disease before a catastrophic event so that they can be treated appropriately. While traditional risk factors for cardiovascular disease currently guide decisions around medical therapy, testing and further intervention are usually indicated by patient-reported symptoms. Inadequate oxygen supply to cardiac muscle causes chest pain. 3 qualities of chest pain that portend cardiac origin include that the pain is substernal or in the center of the chest, that the pain is provoked or exacerbated by physical exertion or emotional stress, and that the pain is relieved by rest or nitroglycerin. In addition to these classic symptoms, shortness of breath can also be considered an anginal equivalent clinically. Anginal symptoms are then classified into one of 3 categories. Patients with all 3 chest pain symptoms have typical angina. 2 symptoms are considered atypical angina, and it's considered nonspecific otherwise. We also have good data that patients experiencing typical angina have a 4-fold increased pre-test probability of atherosclerosis compared to patients with nonspecific angina. However, these data on symptoms are from a clinical trial setting. What we don't know is how reliably these questions are asked in a typical clinical setting and how well they are recorded, despite the fact that the recording of these symptoms in clinical notes are what currently drives guideline-based clinical care decisions. In order to study the predictive value of symptoms as recorded in clinical settings and examine current clinical guidelines that rely on these data, we need to develop and test methods to extract symptoms from clinical notes. Therefore, the purpose of the study was to apply a natural language processing model for the task of extracting angina symptom information from primary care notes. Briefly, the methods we followed were to define a cohort, annotate notes for those patients, apply a language model, and then evaluate the performance of that model. This was a single center study performed at the Providence Veterans Administration Medical Center in Rhode Island. 459 consecutive patients referred for cardiac testing without known atherosclerotic cardiovascular disease by primary care physicians were included. For each patient, the single note from their primary care provider most proximal to cardiac testing referral was selected. The notes were then annotated for 6 questions that align with the information required to calculate pre-test probability of atherosclerosis and determine downstream test appropriateness. 2 annotators were responsible for the annotations of every note. They were provided detailed guidelines as to how to annotate the notes, and those guidelines were used to adjudicate any discrepancies by a 3rd author. A transformer architecture was chosen for this NLP task for 2 reasons. First, they have been shown to perform well on text sequence classification tasks such as this one. And second, compared to traditional NLP methods, the task of labeling the data can be performed by individuals who have domain expertise, in this case, knowledge of the documentation of cardiovascular symptoms, but do not require any knowledge about NLP systems. For this reason, this choice of NLP method is easily scalable to other domains and clinical questions because it decouples the requirement of subject matter from NLP expertise. Bio plus discharge BERT is a language representation model trained on both general and domain-specific text. It has then been fine-tuned on medical text. It is available as an open source Python package and was chosen as a domain-appropriate language model for the task of detecting angina symptoms within clinical notes. We did not have data, prior data to suggest the quality of symptom documentation we would encounter. However, patients referred for stress testing were expected to be enriched for angina symptoms, and the notes should contain answers to the questions we are asking as they are required for downstream test appropriateness protocoling by cardiologists at the Providence VA. Our first valuable finding was that 24% of patients referred for stress testing did not have any information, positive or negative, about chest pain symptoms. Beyond that, a significant portion of notes that mentioned chest pain did not further characterize that pain. This substantially reduced the number of descriptions, both positive and negative, that were available for model training of provocation and palliation of chest pain. Data was split into an 80%, 10%, 10% training, validation, and testing sets. We then performed a 10-fold cross-validation such that all data was represented in a collective validation and testing set exactly once each. The results here represent performance of the collective testing set. We found that we were able to reliably identify mentions of chest pain, substernal chest pain, shortness of breath, and dyspnea on exertion across a range of performance metrics. However, the model performed less well on the two categories of chest pain characterization, provoked by exertion or emotional stress and relieved by rest or nitroglycerin. Our study presents an excellent starting point for characterizing anginal symptoms in clinical notes. While prior studies have extracted chest pain as a symptom from clinical notes, they have had significant limitations. In addition, this is the first study of our knowledge to report NLP extraction of provocation and palliation of chest pain in accordance with cardiovascular medicine guidelines. A strength of this study is that it focused on clinically actionable symptoms that are designed to aid in the diagnosis and determine the pretest probability of coronary artery disease. The answers to these questions impact clinical decision-making, and a refined version of these tools would be deployable for both clinical decision support and to better understand the predictive nature of angina symptoms as recorded in the electronic health record. Our finding that detailed angina symptoms are not consistently recorded in clinical notes for patients who are referred for downstream cardiac testing suggests that we cannot necessarily rely on clinical trial results to inform guidelines for managing chest pain symptoms. It is important that the predictive nature of angina symptoms as recorded in practice be studied. Depending on those findings, it may be necessary to reevaluate guidelines or clinician note-taking practices. Secondly, this study demonstrated that the pre-trained transformer architecture is effective in answering specific clinically actionable questions about patient symptoms when given an adequate training set. It also demonstrated the importance of understanding note-taking practices when designing NLP tasks. I'm now going to briefly highlight two sessions that I attended at AMIA and try to convey just a couple of take-homes that I found particularly useful. The first was a workshop on causality given by Elias Berenbaum at the Center for Causal Research at Columbia. My most significant takeaway from this workshop was an understanding of Judea Pearl's ladder of causality. The image comes from his general audience book, The Book of Why, which I would highly recommend. For a theoretical and mathematical foundation, he also has a textbook on causality. The rungs of this ladder are broken up into three levels. The first is where most health sciences research outside of clinical trials occurs. That is the identification of associations. This answers questions like, what does a symptom tell me about a disease? The second rung is where things get more interesting. These questions tend to be in the domain of clinical trials. If I take an aspirin, will my headache be cured? The third is the world of counterfactuals and are very hard to answer. Was it the aspirin that stopped my headache? We often have data at one rung and we reach for conclusions at a higher one inappropriately. In particular, to answer questions at a layer, we need knowledge at the same layer or higher in order to answer that question. Finally, another interesting sequela of this paradigm is that even the most advanced machine learning technologies rest squarely on level one. At their core, they are extremely good pattern recognizers. That is certainly true for my angina symptom extraction model. Without additional domain knowledge, what Pearl might call a causal model framework, there is no way for these models to answer level 2 questions. The next session I want to highlight briefly was one on data-driven clinical decision support given by Jonathan Chen at Stanford. The best way for me to introduce this was an analogy that he presented to the show, Who Wants to Be a Millionaire? In that show, contestants are asked a series of multiple choice questions and they are given a limited number of lifelines to help them out if they didn't know the answer to a question. One of those is to poll the audience. The technique of crowdsourcing answers turns out to be an extremely powerful one. This ROC curve on the left demonstrates that even if the chance of each individual on, for example, a jury get the answer correct, guilty or not guilty, only slightly better than 50% of the time, a modest jury size quickly gets you to the right answer most of the time. In fact, crowdsourcing answers can often give significantly better results than expert opinion. While there's a lot of noise in the electronic health record, it is a reasonable assumption that the average clinician is better than random in their medical decision-making. Jonathan Chen at Stanford has applied this towards developing and evaluating decision support tools By looking at what is ordered for patients who present with a diagnosis code of chest pain, it is possible to accurately infer treatment guidelines and design appropriate order sets. He hypothesized that this could be useful in informing upstream test ordering. For example, a primary care patient presenting needing a complex referral could be informed as to what tests should be ordered before the appointment of the referral based on how similar referrals have been treated in the past. I'd like to thank my co-authors and funding support for all of this, for this work that I presented today. And feel free to email me if you have any questions in the future. Thank you.
Great, thank you so much, Aaron. So I think we're going to do questions at the end. So just keep adding them to chat and we'll do the ones we can. And then if the speakers want to respond, they can, but just keep them coming in the chat. So I think I'd like to introduce our next speaker. So Jillian is up next. We'll talk a little bit about her AMIA study and some of the highlights from sessions she attended.
Hello, everyone. Today, I would like to share my work titled Phylogenetic Approach to Analyze the Conservativeness of BRCA1 and BRCA2 mutations, which was presented at the AMIA Annual Symposium this year. Breast cancer is one of the most common cancers in women in the US. The 5-year survival rates decline from approximately 100% at stage 1 to about 22% at stage 4. Early detection and prevention of breast cancer can greatly increase the survival rate of breast cancer patients. BRCA1 and BRCA2 are two of the most widely studied genes in breast cancer. Many mutations in BRCA1 and BRCA2 have been found to increase the risk of breast cancer. Both BRCA1 and BRCA2 have a long evolutionary history, which can be dated back to 1.6 billion years ago. BRCA1 is found in plants and animals, while BRCA2 is also present in fungi. Our study proposed the use of a previously developed phylogenetic approach called the Characteristic Attribute Organization System, or CHAOS, to determine the evolutionary conservativeness of different positions for a given gene. The CHAOS method enables identifying Homo sapien pathogenic mutations by just studying the conserved positions in other species. In this study, we proposed the idea of variation number and applied it to BRCA1 and BRCA2 genes to test whether there is a correlation between our phylogenetic measure and the known BRCA1 and BRCA2 pathogenic variants. Let's look at the figure for some definition. We define a character attribute as a state that exists in some or all elements of a clade, but not the alternative clade. For example, for the third position in the left figure, both C and G are character attribute. For the fifth position, only C will be identified as a character attribute. A variation number is defined as the number of occurrences of a position as a character attribute in all the tree clades. The algorithm for calculating the variation number is shown on the right panel. We use a breadth-first search to calculate the variation number for each gene position. We define evolutionary conservativeness based on the previously described variation number. The positions with a relatively small variation number will be considered more conserved than the positions with a larger variation number. In the past, researchers have defined conservativeness based on percentage similarity. Here, we demonstrate a simple example to show why our definition of conservativeness is more advantageous. In the figure, there are two trees. Both of them consist of 3 sequences of cytosine and 2 sequences of adenine, but the sequences are clustered differently. The top clade in the left figure contains all cytosines, while the bottom clade had adenines, suggesting these nucleotides may be conserved for the subgroups. Such pattern is not seen in the figure on the right. The conventional statistical method cannot differentiate the two trees because the percentage similarity in both trees are the same, with 60% of cytosine and 40% of adenine. However, if we assign the variation number to the two trees, the tree on the left will have a variation number of 2, while the tree on the right will have a variation number of 5. Our variation number suggests a different level of conservativeness for the two trees, despite the percentage similarity being the same. We developed a pipeline to calculate variation number and study whether there's correlation between the variation number and disease variants. In order to calculate the variation number for each position of BRCA1 and BRCA2 transcript variants, we first used the basic local alignment tool, also known as BLAST, to find sequences similar to BRCA1 and BRCA2 transcripts. We then aligned the sequences retrieved using muscle software. Afterwards, we generated maximum parsimony tree using POP software. In the end, we calculate the variation number for each position using the breadth-first search algorithm. In parallel, we use CleanVar, which is a public archive of reports of the relationships among human variations and phenotypes to identify BRCA1 and BRCA2 pathogenic positions. We then combine the outputs to perform a t-test by comparing the variation number between the pathogenic positions and the non-pathogenic positions. We found that the phylogenetic trees generated using the BRCA1 or BRCA2 transcripts follow the taxonomy order pattern. Each tree leaf shown in the figure here represents a species. The tree leaves were colored according to the taxonomical order they belong to. Most organisms from the same order were clustered into a single clade, as shown in the figure, suggesting the trees are taxonomically valid, and the BRCA1 and BRCA2 gene evolution follows the species evolution. A t-test was performed to compare the variation number between pathogenic positions and non-pathogenic positions. The pathogenic positions have a mean variation number of 189, while the non-pathogenic positions have a mean variation number of 210. The smaller mean variation number suggests that the pathogenic positions are more conserved compared to the non-pathogenic positions. These results suggest a significant difference between the variation numbers of the pathogenic and non-pathogenic positions, and the pathogenic positions are more conserved than the non-pathogenic positions. A t-test was also performed using the percentage similarity at the pathogenic and non-pathogenic positions. A larger percentage similarity indicates that a given position is more evolutionarily conserved. The t-test results of the BRCA1 BLAST sequence using percentage similarity are shown in this figure. There is no significant difference between the pathogenic and non-pathogenic positions when using the percentage similarity, which further suggests that our variation number is a good measure for the evolutionary conservativeness. In this study, we proposed a novel measure called the variation number, and the smaller variation number suggests more conservativeness. And the mutation occurred at a gene position with relatively small variation number will more likely to be pathogenic. We used CleanVar to identify pathogenic variant positions. We then performed t-tests and found that pathogenic variant positions have smaller variation number compared to non-pathogenic positions. The approach taken in this study alone may not be powerful enough to identify human pathogenic variants. However, it can be used as an important feature for a more sophisticated method in the future. I would also like to share my experience at the Virtual AMIA Annual Symposium this year. So I attended a workshop named Integration of Multi-Scale Multimodal for Clinical Decision Support. The workshop discussed challenges, opportunities, methods, and experience towards integrating multi-scale data towards developing more rigorous clinical decision support tools. The overarching theme focused on the current state of data sets and the methods to bridge diagnostic imaging with clinical and molecular characterization for accurate diagnosis and treatment selection. Topics included extracting and retrieving semantic content from large imaging archives, utilizing image features as part of electronic health record-based phenotyping, applying machine learning to discover the correlation between images and the other biological scales, characterizing the significance of evolutionary features derived from images, and translating multi-scale disease models into practice. The AMIA Annual Symposium has presentations and workshops that include a wide range of topics, which are all tied to health informatics. It's a great experience to have the opportunity to explore what other people are doing in the field and learn how to improve health informatics from different scopes. I would like to thank the following people for guidance and support. Thank you.
Thanks so much, Jillian. So I think we'll go to our next speaker, but if there are any questions, feel free to put them in chat. We'll address them at the end. And we'll now go to Katie.
Thanks, Dr. Chen, and thank you all for being here today. So today I'll be discussing our paper focused on mental health comorbidities seen in pediatric patients with autism. So autism is a group of complex behavioral and developmental disorders that have a broad range of social and emotional clinical presentations. Autism prevalence continues to be on the rise at an estimated 1 in 54 children based on the latest report by the CDC, and there's still a lot unknown about this disorder. Previous studies have indicated that there are certain medical conditions or comorbidities that are more prevalent in children with autism compared to the general pediatric population. And so more focused research is needed, and specifically we focus on mental health conditions diagnosed alongside autism. So here's an overview of our study approach. Medical claims data are a rich source of information for large populations, and Rhode Island is a small state with a large demographic, so it's a great area to obtain a more holistic data set. And we use the Rhode Island All Pairs Claims Database, or HealthFacts RI, for this study. The goal of this study was twofold. First, the study aimed to identify and validate common mental health conditions seen in patients with autism. Most research thus far has only been preliminary with little validation from other large data sources. So by using this large set of medical claims data, trends within the autism population could be tracked and visualized. The second aim of the study was to further analyze and identify patterns in the data by using two unsupervised learning techniques, association rule mining and sequential pattern mining. Diagnosis codes were aggregated and mapped into groupings using a system called Phenome-Wide Association Studies, or PHE-code groupings. This table here shows how diagnosis such as depression can be mapped to the corresponding ICD-9 and ICD-10 codes. As you see, there are many of them. And we also chose 38 PHE-code mental health groupings as the focus of the study, and some of them are listed here on this slide. Association rule mining is a well-established method that is useful for identifying correlations and patterns of items in the data set. The classic example of this method is with the market basket analysis, which uncovers items that are bought together at a grocery store, such as the unexpected association of diapers and beer. In turn, the grocery store might put these items closer in proximity to one another, or order ones that could be in close association with one another. Association rule mining can also be used in other fields such as healthcare. This study used it to identify frequent combinations of mental health conditions unique to the autism population. And in this case, the item set included all of the mental health FreeWAS or PHE-code groupings chosen for the study. And from this item set, each patient had a set of mental health diagnoses or transactions. Association rule mining does not consider any type of sequence in which these items are purchased, but sequential pattern mining does, and that's the second method used in this study. Sequential pattern mining was originally used in retail to determine the order of items purchased sequentially over time. So in this case of my study, items were ordered based on a list of each patient and their corresponding diagnoses in sequence based on the age that they first appeared in the data set. So now to talk a little bit about the results of the study. First, we looked at some initial descriptive statistics of the autism population within the HealthFacts RI dataset, which were consistent with the national CDC trends, as I mentioned in the introductory slide. And listed here are some of the top mental health disorders where the prevalence of autism population is compared to the general pediatric population. As you can see, a majority of the disorders have a higher prevalence in the autism population for this data set. Association rule mining was performed to get the top rules for the entire autism population, as well as gender and age groupings. Here, for today's talk, I'm just going to talk about the entire autism population. And this was done by removing any rules seen for the entire general pediatric population, which left rules unique only to the autism population. The chi-square metric was used to rank these rules. And as you can see here, suicidal ideation diagnosis was commonly seen in combination with ADHD, anxiety, conduct disorders, and mood disorder. Further clinician validation and analysis is needed to understand why these patterns and combinations of conditions are seen frequently together and maybe not independent of one another, with a specific concentration on suicidal ideation, mood disorders, and conduct disorders. For a more visual representation of the rules, the top rules here can be seen with a graph-based visualization where item sets are represented with the vertices. Arrows pointing from items to rule vertices indicates a left-hand side item, and the arrow from a rule to an item indicates right-hand side. Interest measures are typically added to the plot using color and size, as you can see. And so in this case, it's chi-square statistic and the support cutoff. These innermost conditions here are on the plot are the ones frequently in combination with one another, which is what I discussed on the previous slide. Now into sequential pattern mining. Our analysis identified ordered sequences of mental health disorders in the data set. For example, the top sequential pattern mining rules indicated that combinations of disorders resulted with the right-hand side rule of ADHD. And this is a very common condition seen with autism patients, and it's validated in the literature, so this made sense. It is also important to note that, as with association rule mining, a right-handed side rule of suicidal ideation was observed several times throughout the top 100 rules, so future work is needed to understand the risk for suicidal ideation and attempt and self-harm behaviors unique to this patient population. So in conclusion, limited studies have focused on the studying of the progression of autism. This study validates that mental health comorbidities in children with autism have a higher prevalence compared to the general pediatric population. By applying methods such as association rule mining and sequential pattern mining to these medical claims data set, this study characterized combinations and sequences of mental health disorders associated with the disorder. Based on the observed patterns and temporal sequences, the following mental health disorders need focused attention: suicidal ideation, mood disorders, anxiety, and conduct disorders. This study unveiled important conditions to focus on so that interventions can be developed to improve patient outcomes. This figure on the right shows a patient with several diagnoses and common patterns of conditions seen that may lead to a safety concern, such as suicidal ideation. This figure shows how there's other safety concerns that might be prevalent in the autism population, and future work is needed to stratify this risk and develop predictive models for specific mental health disorders and safety outcomes. So now to talk a little bit about the AMIA 2020 Virtual Annual Symposium. Even though we didn't get to go to AMIA 2020 in person this year in Chicago, it was still a lot of fun, and the virtual format had a lot to offer. I was pleased with the chats. They were very lively, and there were many thoughtful Q&A sessions at the end of each talk, which I was pleased about in a virtual setting. I was able to engage and also learn so much by attending many sessions throughout the week. And it was an honor to participate in the student paper competition with students across the nation. So I'll briefly talk about three different sessions that I went to. The first day of the conference, I was able to attend a workshop entitled The New Digital Age of Clinical Decision Support Tools. I have a particular interest in Smart on Fire applications, and this workshop was a great one to attend to just get more familiar with some of the technologies used to create these apps and from end to end and using FHIR standards. For those unfamiliar with Smart on Fire is basically an app store for electronic health records. It requires the use of the same standards so that the application can be disseminated and used by different facilities and EHRs across the nation, which has been a huge problem ever since the adoption, the large adoption of electronic health records. And implementing the interface in a FHIR format enables to have simplified and secure access to patient data. So in this workshop, we worked on a virtual machine to create a clinical decision support tool in a sandbox. and to aid providers in choosing a statin therapy. And it was a lot of fun. So to keep the theme going, I also enjoyed attending the AMIA Fire app competition. It's a popular event at AMIA for the past 3 years to learn about the cutting edge fire applications that are currently in practice. So it showcases these apps that are already in use by health systems. And the first through third place winners that were announced at the end of the conference are listed here. And the first place winner well sheet is a which shows the app on the right-hand side of this slide, is a predictive model application integrated into EHR systems, where the problem they're trying to solve is there's EHR design flaws that impede physician workflow and obscure clinical information. And so it's important to try to solve this problem that costs billions of dollars a year to our healthcare system. So they embedded the technology through Smart on Fire protocols to gain access to patient information in the chart, and then use predictive models to surface the most relevant content and decision support resources for the physician, to also save time and energy by the clinicians. And lastly, just to talk a little bit about the a panel that was entitled, well, basically it was talking about AI and machine learning techniques, and how a lot of these AI algorithms that are proposed in literature are actually not being implemented into practice. And so a group of multidisciplinary panelists talked about how we can standardize and quicken up this process for better and faster implementation of these algorithms into practice. So with that, I'd like to thank you. It was a lot of fun, and I enjoyed the symposium a lot, and I look forward to it each year.
Great. Thank you so much, Katie. And I think Katie mentioned she participated in student paper competition, but she didn't mention that she actually won third place. So so congratulate her for that. Okay, and we'll go to our last speaker, Ryan.
Good afternoon. Happy Friday. Thank you for being here. Thank you for the introduction, Dr. Chen. I appreciate the platform to share. So my name is Ryan Buckland. I was a master's student working in Dr. Chen's lab. And one of the papers that came out of that I presented at AMIA is this one, the selection of clinical text features for classifying suicide attempts. There we go. So just some quick background. I presented this at one of the sessions on mental health informatics. Basically, there's a couple of different types of problems that drive some challenges around classifying suicide attempts. Obviously, there's a public health problem. Suicide is actually shown to be driving life expectancy down. This is a quick screenshot of the CDC director's statement back in 2018, indicating that life expectancy is being driven down by these kinds of things, drug overdose and suicide. It's also a clinical workflow and methods challenge, being able to address and treat folks with suicidal thoughts and behaviors. So there have been several studies that have shown via retrospective chart reviews that anywhere between 70 and 95% of patients with STBs documented in their chart were missing the requisite ICD codes. And you can imagine this has cascading effects both in clinical treatment and clinical decision support, as well as research and some cohort definition steps in empirical studies. The solution that I was most interested in was utilizing clinical text to support this patient identification. Doing some quick literature review and some more in-depth literature review showed that existing approaches were either too manual or too technical to, I think, really be well implemented in your typical hospital system. So from that, the objective for the study was to test an accessible and computational approach that processes this text and uses it to classify specifically patient overdose encounters as STBs or not. So the data source that I ended up using is something that I think folks probably are particularly familiar with. It's the Medical Information Mart for Intensive Care out of Beth Israel Deaconess. It's about 60,000 plus and growing ICU admissions between 2001 and 2012. And then I alluded to this earlier, my kind of interest was admissions of patients with a non-fatal overdose or suicide attempt by poisoning, just because some of the domain literature indicates that there's some questions around intentionality in this group of patients. The data specifically that I pulled from MIMIC was diagnosis codes and discharge summaries. This table at the bottom here shows essentially what I used to label and why I pulled the diagnosis codes. So you can see there those specific IC codes that map to not having a kind of overdose driven by STBs, and those that kind of map to actually being marked positively as having an overdose driven by STBs. So one of the kind of big challenges for kind of doing this in an accessible way is picking a text processing kind of approach. And I chose MetaMap, both because of its availability at BCVI and its kind of publicly accessible format. So this program essentially maps biomedical text to concepts in the unified medical language system, Metathesaurus. You can think of it as a thesaurus essentially, right? And each concept has a unique identifier, aptly called a concept unique identifier. So we can see with this really kind of basic schematic here, what the underlying process is really meant to achieve. So we've got the database of these overdose admits. I pulled all the discharge summaries attached to these encounters, run them through MetaMap program, and get fielded output. That's kind of an intermediate step that I parse some more so I can get what we are particularly familiar with, which is kind of more structured data, essentially a one-hot encoded approach to the appearance of QEs in discharge summaries. So as I mentioned just now, I parsed this output into a one-hot encoded data set of QEs, where each QE was a binary feature indicated by 1 if it was present in the discharge summary for a given hospital admission, or 0 if it wasn't. For the sake of kind of simplicity, I kind of added a couple more criteria based on the MetaMap output. The concept was not to be negated, and I also did some kind of domain-oriented filtering on semantic type. This table here shows the breakdown of QEs by semantic type. So you can see the finding semantic type represented over half of the QEs, and that's something I'll speak to in just a minute, but the rest of the semantic types you can think of as kind of categories that are clinically relevant, and these kind of pop out as what you might think would be relevant to my problem, mental or behavioral dysfunction, mental process, and so on and so forth. So this is kind of the methods that I kind of approach from a classification standpoint. I ended up subsetting my data of one-hot encoded QEs by the semantic type. So I alluded to just in the previous slide, there's this finding semantic type, which I included because it includes concepts like suicide, literally, but it also includes a ton of other things that were not kind of relevant to my problem. So I was interested in playing around with what would the impact be if I took that out of my data set. And so I did this kind of full data set where the predictors were 5,866 different features. And then once I netted out that finding data type, the reduced data set was 2709. And I performed elastic net logistic regression on these two data sets, of course, training and testing split beforehand, and used the features that came out of that process to predict in a second logistic regression whether or not a patient encounter was suicidal thoughts and behaviors driven overdose or not. This is quickly some results. So you can see I was able to reduce the kind of feature space significantly. You can see some in the full data set that are kind of reminiscent of what you would think. So suicide, history of depression. You also see some that don't really have an immediate conceptual connection, like 4+ answer the question, or PSA level less than 5. And those are some of that kind of additional noise that gets pulled in. And then you can see kind of in the subset, the reduced features that were selected, also interesting, like psychiatric hospitalization and so on and so forth. These are the results. So I was curious to test what the performance difference between my full model and my reduced model might look like. Interestingly, it did not affect it too severely. I still was, given the approach I took, pleasantly surprised and pleased by some of these results, which you can see there generally are good. In conclusion, I think it's important to kind of just recognize that the primary goal was to just identify Kiwis for inclusion in a more comprehensive model, not just have an exclusively Kiwi-driven model for this kind of classification approach. The work demonstrated that you can have an accessible and computational approach kind of simultaneously. Definitely future work exists. So you'll notice that my labeling was done by ICD codes. There's absolutely some work around gold standard outcome labels by clinician chart review. Also looking at a different type of clinical document, I chose discharge summary just because it is in fact a summary and quite long relative to other notes. And I just thought it would be a good place to start. Obviously incorporating structured data like I mentioned, and then some other tuning and looking at other ML approaches. Really quickly, my AMIA experience, I am currently at Lifespan as a data scientist supporting the pharmacy organization. So one of the things that I focused on when I was looking at things to attend were things that were relevant to that. So I attended this session on building translational hospital data science teams, leaders from New York University Health System, and then as well as Ochsner in Louisiana, and I think a couple of other states, as well as University of Pennsylvania, all kind of shared how they've built their data science teams, which was particularly interesting to me, just given that I'm the first data scientist in the pharmacy organization that Lifespan has hired. And then, of course, I can't not mention the fact that my session that I presented at ended up actually surprisingly being moderated by the director of NLM, who I think has a background in psychiatric nursing. So that was really interesting. And then finally, just because I'm currently kind of a full-time employee, the fact that the virtual format means that I can go back and review content that I wanted to, but I was working kind of after the fact recordings is particularly helpful. And with that, that's it. I would like to acknowledge, of course, my co-authors, Dr. Chen and Dr. Hogan in the Biostats department, and then, of course, my funding source. Okay. And please feel free to reach out if you have questions.
Great, thank you so much. Perfect timing for everyone. So just wanted to thank the speakers. As you've heard, the conference was virtual, so it's basically recorded talk. So this was the opportunity to do them live. So appreciate you joining us for that, to kind of hear them do the abbreviated versions of their talks. And hopefully you also enjoyed hearing a little bit about kind of other things that they saw at the conference. I think we have time for maybe one question. I see that there's one in chat that's been answered. Are there any other questions from anyone about any of the talks, or just about AMIA or anything in general? And I don't know, Aaron, if you wanted, I think if all the speakers, you can turn your video on. I mean, if you want to kind of elaborate on the question that was asked, since we have a little bit of time.
Sure. So Dr. Fraser asked whether or not there were any other notes that would contain relevant chest pain information that we may have been able to look at in addition to the most proximal primary care note. And so the data that we used for this study was part of actually a larger study where we did a chart review to look for this information. And overwhelmingly, the information, if available, was in the most proximal PCP notes. Kind of the workflow is that patient comes to their primary care doctor, they have a complaint of chest pain or related symptoms, and then the decision gets made to be referred for cardiac testing. So really that's where the information should be documented. In some cases, there was additional information, although usually it overlapped with the information in the primary care note for a recent emergency department visit where a patient went to the emergency department, had chest pain, they ruled out for a STEMI, but then were suggested that they should go to their primary care doctor for further workup. But again, in most cases, the information in those two notes overlapped.
Thanks, Aaron. That was really helpful. And I think it underlines one of those unfortunate secrets about electronic health record notes, that clinicians don't always document really important stuff. And there are lots of longer conversations as to why that is, but it's definitely unfortunate when you have such key symptoms for something like angina, and that's not information not being recorded in the chart.
Great. Any other thoughts or questions? Okay, well, feel free to reach out to our speakers. I'm gonna hand it over to Tara for some closing thoughts. She's done basically the behind the scenes, so really appreciate everything that she's done. Really glad the speakers had the chance, really showcasing the work that we're doing in BCBI and CMB, and also showcasing some of the data we have access to, the computational resources. So thank you to the speakers. Thank you to all of you who attended. It's nice to see some new names join our community. We look forward to seeing you more in the new year. And then just wanted to hand over quickly to Tara to kind of close out our session and our year in review.
Thank you, Liz. Yes, I just wanted to just echo what Dr. Chen said. We are really trying to, reach out to everyone and build community, and this format has been great to hear from all of our different speakers. So we encourage you to continue joining us. Next semester, we've got a lot planned. So thank you for being here. Have a safe and happy winter break, and we'll see you in 2021.
Thanks, Tara.
Thank you.
Have a great holiday.
Happy holidays, everyone.